Welcome back to the video series on the detailed solution explanation video of 2014 M.Sc Botany Entrance Exam of Delhi University. If you want the PDF of this question paper, I will give you the authentic link in the description box of this video. So let's start with the first question. Zymogen is N. So there are 4 options given. So let's see what actually is a zymogen. So a zymogen is an enzyme precursor which is inactive and often called as proenzyme. A zymogen become active either by some environmental factor, for example heat stress, or through some biochemical changes, for example hydrolysis, etc., due to which its inhibitory part is cleaved or its active sites are exposed. So the biochemical alteration of zymogen, which is actually a proenzyme, to active enzyme usually occur in the Golgi bodies. So you can see, for example, how the proinsulin, which is a proenzyme or a zymogen we can call it, is converted to active insulin by the removal of a 33 amino acid long peptide chain. So the answer to this question will be that zymogen is an enzyme precursor. Next question, hydrolysis of fats by alkalis into fatty acid and glycerol is called as. The options are suspension, colloidal, coagulation, or saponification. So saponification is a process that involves the conversion of fat, oil, or lipid into fatty acid and corresponding alcohol by the action of a strong aqueous alkali, for example NaOH, in the presence of heat. You can see the reaction in the image below. So the option D, that is saponification, is correct. Beta oxidation takes place in. The options are chloroplast, cytoplasm, mitochondria, ribosomes. So let's see what is a beta oxidation. So beta oxidation is the catabolism of fatty acid to generate acetyl-CoA, NADH, and FADH2. It occurs in cytosol in case of prokaryotes because they do not have membrane-bound organelles, and mitochondria in case of eukaryotes. It may also occur in peroxisomes if very long chain fatty acids are involved for oxidation. The acetyl-CoA then goes to the Krebs cycle, and NADH and FADH2 coenzyme goes to be used in the ETC. The beta oxidation is so named since it involves the oxidation of the beta carbon of the fatty acid into a carbonyl group. The enzyme for beta oxidation are present in the inner membrane of mitochondria in eukaryotes. So the option C, that is beta oxidation takes place in the mitochondria, is correct. Next question, occurrence of anther and stigma at different levels in a flower is referred as hercogamy, dichogamy, flower constancy, or protandry. We will define each of the following one by one. So hercogamy is the occurrence of anthers and the stigma at different levels in a flower and is a common strategy employed by bisexual angiosperms to reduce sexual interference within and promote cross-pollination. There are 2 types of hercogamy. The approach hercogamy, in which the stigma is present above the level of anthers, and reverse hercogamy, in which the stigma is present below the level of anthers. Dichogamy refers to the maturation of anther and stigma of the same flowers at different time. It promotes cross-pollination even in the bisexual species, and it is of 2 types. Protogyny, when pistil matures before anther, for example pearl millet, and protandry, when anther, that is pollen grain, mature before pistil, for example maize, sugar beet, etc. So dichogamy and hercogamy are the phenomena to prevent the self pollination, but the thing is that hercogamy is the separation of 2 sexes spatially, means they are located at different position, while dichogamy is the separation of sexes due to the time because they mature at different stages. And flower constancy is the tendency of pollinators to specifically visit particular flower species and not visiting other flower species in the vicinity, even if those flowers are more rewarding, that is they contain more nectar. So the answer to this question will be hercogamy, that is occurrence of anther and the stigma at different level in flower is referred as hercogamy. Next question, malacophily refers to pollination by. So this is very straightforward. question, the malacophaly is a pollination of plants by snails and slugs. So the option A, that is malacophaly, refers to the pollination by snails and slugs, is correct. Next question, the growth pattern in pollen tube is similar to that of fungal hyphae or root hairs in exhibiting. So the pollen tube was first discovered by G.B. Amici in Portulaca oleracea. The tube elongates with the elongation of the cytoskeleton and extends from the tip. The extension is influenced by calcium. The calcium assists synaptic vesicle growth and extension at the tip. In addition, some small peptides are also released by the synergids, which attracts the pollen tube to grow towards the embryo sac. So the answer to this question will be that the growth pattern in pollen tube is similar to that of fungal hyphae or root hairs in exhibiting the tip oriented growth. In the description box of this video, I have given a link for a very good review which gives a exhaustive detail on the pollen tube growth, and it has very good illustrative figures also. You can go to the link and see read that review if you are interested. However, this review is a very exhaustive review, so it won't be helpful in your MSc entrance exam. So I will advise you to read this review whenever you are free or you have free time. Coming to next question, the pollen tube attraction towards the embryo sac is mediated by small peptides produced from the. So this explanation I have already given in the previous slide. So the answer is synergids. Next question. One of the essential prerequisites for normal microgametogenesis to proceed is. So microspore is the first cell of the male gametophyte and is haploid, and during early stage of microgametogenesis, they undergo unequal division, becomes vacuolated and forms a small generative cell and a large vegetative or tube cell. So the answer to this question will be second, that is one of the essential prerequisites for normal microgametogenesis to proceed is vacuole formation and asymmetric or unequal division in the microspores. Now the question is, choose the incorrect statement about three-celled pollen grain. So let's see the characteristic of three-celled pollen grains. The three-celled pollen grains are generally short-lived because their respiration rate is high. The SIS, that is self-incompatible species, is common with three-celled pollen grains and is often found in the members of the mustard family, that is brassica, including turnip, cabbage, cauliflower, etc. And trinucleate pollen grain is very hard to germinate on artificial media, and binucleate one is easy to handle and germinate. So from the first point itself, we know that the first option, that is they are generally long-lived, is incorrect, and this is the answer to our question. Coming to next question, RNase mediated inhibition of pollen tube leading to its inhibition in the pistil conforms to. Now let's see what is self-incompatibility. Self-incompatibility is a general name for several genetic mechanisms in angiosperms which prevent self-fertilization and thus encourage outcross and allogamy. So best studied mechanism of self-incompatibility act by inhibiting the germination of pollen on stigmas or the elongation of the pollen tube in the styles. So this self-incompatibility is very huge topic, and there are various types of self-incompatibility. I have given a link in the description box. You can go to that link and read about all the types of self-incompatibility, and it is very important also. So I will just discuss here the gametophytic self-incompatibility. This is the most common type of self-incompatibility, and it occurs through two different mechanisms at molecular level, the RNase mechanism and the S-glycoprotein mechanism. In the RNase mechanism, when the pollen tube reaches around 1/3 of the style length, its growth is inhibited by an S-locus encoded ribonuclease, which is called as sRNase, which is synthesized within the style, and it enters the pollen tube and destroys the rRNA, that is the ribosomal RNA molecule in the pollen tube, due to which the pollen tube elongation is arrested and the pollen grain dies. So the answer to this question will be that RNase mediated inhibition of pollen tube leading to its inhibition in the pistil conforms to gametophytic self-incompatibility. Next question, cellular endosperm formation is characterized by. So there are three types of endosperm. The first one is nuclear endosperm, in which the primary endosperm nucleus divides without wall formation. It is the most common type. For example, it is found in cotton, maize, etc. The second one is cellular endosperm, in which there is no free nuclear division, and every division of primary endosperm nucleus is followed by wall formation. For example, it is found in Datura and Petunia, etc. And then halobial type of endosperm. It is found in members of the order Halobiales, and halobial endosperm is intermediate between the nuclear and the cellular types. The first division of primary endosperm nucleus is followed by a transfer cytokinesis resulting in micropylar and chelazal chamber, and further development in both the chamber is of free nuclear type. So the answer to this question will be that cellular endosperm formation is characterized by absence of free nuclear division. Next question, transfer cells are involved in. So you can see the figure on the right hand side in which the transfer cells are shown. The transfer cells are specialized parenchyma cells having plasma membrane enfoldings and invaginated secondary walls for increasing the surface area. You can see the invaginated wall in the figure also, where the transfer cells are shown on the rightmost side. They facilitate the high rate short distance apo or symplastic transport of assimilate from sugar source, for example leaves, etc. And the term transfer cell was coined by B.E.S. Gunning and J.S. Pate. So the option A, that is transfer cells are involved in short distance transport of assimilate. Next question, central thicker part of a bordered pit membrane is called. So pits are relatively thinner areas of the cell walls with secondary layers. They occur in pairs, often called pit pairs, through which two adjacent cells can communicate. And there are two types of pits, the simple pits, a pit pair which have equal diameter of the pit chamber and aperture, and bordered pits, a pit pair with over-arched pit chamber, a smaller pit aperture. So you can see in the bordered pit that the pit chamber is over-arched and there is a smaller pit aperture, and in the middle you can see the thick or bulged part of the pit membrane, which is called as torus. So the answer to this question will be that a central thicker part of a bordered pit membrane is called as torus. Next question, stomata in which the common wall of the two subsidiary cells is at right angle to the guard cells is called. So these are the four stomata type which are our options. So you can see that on the left hand side is the N-isocytic type, in which the three subsidiary cells surround the guard cell and one subsidiary cell is smaller than the other two. On the topmost you can see the cyclocytic type, in which four or more subsidiary cells surround the guard cell inner ring. On the downside you can see the diacytic type, in which the subsidiary cell are at right angles to the guard cell, and on the rightmost you can see the tetracytic type, in which the four subsidiary cells surround the guard cell. So the answer to this question will be diacytic type, because you can see in the figure that in the diacytic types the subsidiary cells are at right angle to the guard cells. Next question, cork cells are impervious to water and air because their walls are impregnated with. So cork cells are dead, heavily thickened and impregnated with an organic substance, suberin. Suberin makes these cells impervious to water and gases. On the right hand side you can see the very famous image from the book Micrographia, written by Robert Hooke in 1665, when he was postulating his cell theory, and he saw these cork cells through his microscope. So the answer to this will be that cork cells are impervious to water and air because their walls are impregnated with suberin. So in the next video I will be dealing with the detailed solution and explanation of question number 91 to question number 105. If you have any doubt in the solution, please do comment. Please like the video and do comment and subscribe to appreciate my work, and stay tuned for more upcoming solution videos of various M.Sc entrance exams related to biological sciences.